Okay, good morning, good afternoon, good evening. Welcome everyone today to Global IMMUNOTALKs. Global IMMUNOTALKs are an initiative created in 2020 to spread immunology and inspire the new generation of scientists all around the world in a friendly and egalitarian manner. I am Gabriel Rabinovich, and I have the honor and privilege of presenting our speaker today, Dr. Karina Bortoluci. Karina has finished her graduate studies in 1995 at the Mackenzie University in Brazil, and performed her PhD and postdoctoral training at the University of Sao Paulo, Brazil. She was the head of the Department of Biological Sciences at the Federal University of Sao Paulo in 2002-2003, and was a sabbatical visiting scientist at Genentech in San Francisco in Dixit Lab in 2016. She has been honored by CNPq, the Brazilian Council of Scientific and Technological Development, as one of the 22 outstanding young scientists in Brazil. Currently, she's associate professor at the Department of Pharmacology at the Federal University of Sao Paulo. Karina is a specialist in the field of innate immunity. She has made outstanding pioneering discoveries on the role of inflammasomes during infectious and inflammatory diseases, and has published seminal papers in this field, including those reported in PNAS, Immunity, Journal of Biological Chemistry, and Nature Medicine. Karina is also deeply involved in mentoring graduate students and actively engaging in extension activities, including organization of symposia, courses, and workshops. In a personal note, I would like to say that I am particularly touched about presenting Karina today. Both of us are based in South America. In South American countries. Karina is from Brazil, I am from Argentina, and I know exactly how challenging it is to accomplish experimental research and do mentoring in our countries. So I have a strong appreciation for Karina's work and have a lot of admiration for her, not only professionally, but also personally. Karina will talk today on how do inflammasomes shape astrocytes' ability to control infection. But before you start your talk, Karina, one of the traditions of Global IMMUNOTALKs is to ask our speakers a special question. In this case, the question for you is, what is your mentorship style? Was it always like that, or did it evolve over time? Please.
So thank you. Good morning or good afternoon, everybody. And thanks for the invitation. Thank you, Gabi, for this warm introduction. Yes, we are good friends, and I'm very happy you introduced me. So in regards to my mentorship, and I absolutely adore talking about mentorship because it's truly my passion and where I devote most of my time and energy as a PI. To me, mentorship goes beyond just offering scientific or technical guidance to my students. It's more about nurturing their holistic development, including fostering their self-awareness. I love working with the diverse personalities, and I always encourage them, my students, to explore their particular motivations, values, unique talents, and abilities. And as a strategy, besides our weekly lab meetings and journal clubs, we use it to hold brainstorm sessions every 3-4 months to assess the goals they achieved, like that picture there, which steps they climbed, and of course, why some may not have worked out. which helps them gain greater clarity on their professional path. Furthermore, I think I place a strong emphasis on cultivating future scientists who are capable of asking insightful questions rather than merely providing answers. And I don't think my mentorship style hasn't changed a lot over time, but I've definitely matured and become more myself, self-aware. And I think this has enabled me to guide my mentees in gaining a deeper understanding of their career aspirations beyond just completing their thesis or getting published. So I think my ultimate goal is to instill in them a sense of joy in the process itself, not just the end result. I think it is it.
Much, Karina. This is fantastic. Thanks for inspiring words. And now I would like to invite you to share the screen and deliver your lecture. We are very much looking forward to it.
Okay, thank you. That's fine?
That's perfect.
Okay, so let's go. So today I will talk about how inflammasomes shape astrocytes' ability to control infections. But before we delve into the talk, let's quickly recap. Let's just quickly recap what conventional inflammasomes are. So we know that these canonical inflammasomes are multi-protein platforms present on the cell cytoplasm of many cell types, and they activate caspase-1, the inflammatory caspase-1. These platforms are initiated by sensor proteins from node-like receptors or PHIN families that interact to procaspase-1 directly or through the adaptor molecule ASP. Stimuli given by pathogen or damage-associated molecular patterns or also homeostasis-altering molecular patterns induce a conformational change in the sensor protein, leading to the assembly of the platform. And the recruitment of caspase-1 into this scaffold-like structure results in the self-cleavage and maturation of caspase-1. Active caspase-1 then cleaves its substrates, pro-IL-1 beta, pro-IL-18, into their mature forms, and also cleaves this protein named gazermin D. The N-terminals of the cleaved gazermin D inserts into the cell membrane, leading to a pore formation by which these cytokines can be released and potentially giving rise to a lytic and inflammatory process of cell death named parapitosis. So traditionally, the effector mechanisms mediated by inflammasomes are cytokine release and the induction of parapitosis. However, it is important to note that the cell death is not always the final outcome of inflammasome activation. Some cells don't die and become hyperactive, secreting high levels of these inflammatory cytokines. Moreover, these are not the only effector mechanisms mediated by inflammasomes. Recently, it has been described many effector responses mediated by inflammasomes, such as secretion of inflammatory mediators and microbicidal molecules, cell-specific effector mechanism to control infections, processes of cell death, and so on. In this sense, our group has been contributing to describe novel effector mechanisms mediated by inflammasomes. For instance, we described a pathway for inducible nitric oxide synthase activation that depends on caspase-1, but it is independent on IL-1 beta and IL-18. And it involves a epigenetic regulation through the PARP1 cleavage. And the consequent nitric oxide production through this pathway is important to the macrophage ability to fight protozoan and bacterial infections. Moreover, we described autophagy as another effector mechanism mediated by inflammasomes to macrophage control of infections. And finally, we also demonstrated a lysosomal pathway induced by inflammasome that are responsible for a peculiar form of cell death and also secretion of inflammatory mediators involved also in the control of infections. So together, these finds demonstrate the multifaceted nature of inflammasomes and highlights the action of these platforms to induce effector responses beyond this conventional one. Moreover, the presence and impact of inflammasomes on non-immune cells highlights the importance to study the inflammasome biology in many cell types, across many cell types, with what we are doing now in our lab, focusing on these non-hematopoietic cells, the astrocytes. So astrocytes are the most abundant non-neuronal cells present in the central nervous system, where they play a crucial role for the brain homeostasis. So we know that astrocytes has an important role in maintaining the metabolic support for neurons, and also maintaining the integrity of brain-blood barrier. Moreover, astrocytes respond to infections by secreting inflammatory mediators. And they also express many pattern recognition receptors and can be directly or indirectly activated by infectious agents. However, the autonomous ability of astrocytes to control infections remains elusive. We know that in addition to microglia that are considered the macrophage of the central nervous system, other brain cells, including astrocytes, express many components of inflammasome. However, we don't know yet how inflammasomes are really activated on these cells and the impact of this activation. We know that, similar as described for immune cells, inflammasomes has dual role on the central nervous system, the Bodica and the yoga roles. So in addition to the well-known role of inflammasomes in neurodegenerative diseases, mental disorders, and neuroinflammation, inflammasomes has also physiological and protective roles in this compartment, such as participating during neurodevelopment, being protective in the late phase of experimental autoimmune encephalomyelitis, and also being involved in controlling infections in this compartment. However, the role of inflammasomes on the ability of astrocytes to control infections remains to be elucidated. So to answer this question, how inflammasomes are activated at the end, the impact of this activation for astrocytes control of infections, we use it as approach the infection with the two viruses that are known to reach the central nervous system, Zika virus and SARS-CoV-2. Zika virus is a flavivirus that is transmitted by the bite of Anopheles mosquito, but can be also transmitted through the sexual contacts or from mother to fetus. In 2016, Zika virus gained attention because of these devastating neurological consequences of this virus, such as microcephaly, Guillain-Barré syndromes, encephalomyelitis, and so on. On the other hand, SARS-CoV-2 is primarily a respiratory virus, but we know that this virus can reach the brain and by routes that are not completely understood, and then we know that virus can cause some pathologies. Great proportion of COVID patients present neurological consequences. And more recently, a group of Brazilian friends demonstrated that astrocytes can be an important site for SARS-CoV-2 replication in the central nervous system. And in regards of inflammasome activation in response to Zika virus and SARS-CoV-2, we know that inflammasomes has gained attention in response to COVID, especially in the context of lungs and monocytes from COVID patients. The activation of an NLRP3 inflammasome has been related to the disease severity. And more recently, it was demonstrated that NLRP3 inflammasome are also found in microglia from COVID patients. In response to Zika virus, the consequence of inflammasome activation is still controversial because we can find protective roles for inflammasome in controlling infection, but also we can find some detrimental roles in the literature. In regards to the activation of inflammasome in response to Zika virus in glial cells, it is still more controversial because it was found that in a human microglial cell line, Zika virus can induce inflammasome activation and IL-1 beta secretion. However, in a mixed glial cell culture, Zika virus not only fail to induce inflammasome activation, but also inhibit the activation in response to the classical inflammasome agonists. So in order to better understand how these viruses can activate inflammasomes and the impact of this activation to the control of infection, we first infected primary astrocytes obtained from the cortices from newborn mice with the Brazilian strain of Zika virus and also with the P1 gummy strain from SARS-CoV-2. And we observed that both viruses were able to invade astrocytes and replicate within those cells. Zika virus replicate across time, and the peak of infection in response to SARS-CoV-2 was found 48 hours after infection. After this time, we observed a clear reduction in the viral load, just suggesting that astrocytes can control SARS-CoV-2 infection. Interestingly, both Zika virus and SARS-CoV-2 were able to induce caspase-1 activation in astrocytes, similar than observed for nigericin, that is a classical NLRP3 inflammasome agonist. And most importantly, caspase-1 seems to be required for the control of these viruses by astrocytes, since higher viral load was observed in astrocytes lacking caspase-1 for Zika virus and for SARS-CoV-2, a trend consistent with our immunofluorescence observation. Here we can see much more Zika virus intracellularly, as well as SARS-CoV-2 in the absence of caspase-1. But interestingly, only SARS-CoV-2, but not Zika virus, were able to activate other classical inflammasome activation signals on astrocytes, such as IL-1 beta and also ASC-SPEC formation. ASC-SPEC formation is a signal of inflammasome assembly, and we can only found it on astrocytes in response to SARS-CoV-2, but not in response to Zika virus, thus suggesting that only SARS-CoV-2 was able to induce a classical inflammasome activation on astrocytes. So we delved further to investigate this activation of inflammasomes and the impact to SARS-CoV-2, to the control of SARS-CoV-2 infection by astrocytes. First, we observed that ASC-SPEC formation, caspase-1 activation, and IL-1 beta secretion were lost in the absence of NLRP3 and caspase-1. So indicating that SARS-CoV-2 activates NLRP3 inflammasomes on astrocytes, similar as described for human and murine macrophages and monocytes. To further understand how SARS-CoV-2 activates NLRP3 inflammasomes on astrocytes, we transfected those cells with the spike and nucleocapsid proteins. And we observed a clear induction of ASC-SPEC formation in response to both proteins, and also IL-1 beta secretion in response to N and S proteins, in contrast to that observed for flagellin, a non-related protein and an agonist of other inflammasome, the NAPE and LRC4 inflammasome. And as expected, again, these responses were lost in astrocytes from NLRP3 and caspase-1 null cells, similar as we observed for nigericin. Here, no response in the absence of NLRP3 and ASC in response to N protein, and also in response to S protein. So indicating that SARS-CoV-2, the activation of NLRP3 by SARS-CoV-2 relies on N and S protein from SARS-CoV-2. So just to remind you, different from the majority of pattern recognition receptors that are activated by the direct binding to their cognate ligand, NLRP3 seems to be indirectly activated by many molecular distinct stimuli. These stimuli seems to induce common cytosolic pathways that are responsible to activate NLRP3. And these common pathways involves potential efflux, lysosomal disruption with the cathepsin release, and the release of ROS from mitochondria. And accordingly, the inhibition of these common pathways significantly reduced IL-1 beta secretion in response to nigericin, and also abrogated IL-1 beta secretion in response to the nucleocapsid and spike protein from the virus. So thus indicating a classical NLRP3 activation in response to SARS-CoV-2 in astrocytes. So next, we want to understand what's the impact of NLRP3 activation on astrocytes for the control of this infection. And we observed higher viral loads in NLRP3 and caspase-1 new astrocytes, but not in the NLRC4 new astrocytes, in supernatant as well as in lysate, cell lysate. A trend again consistent with the observation of intracellular virus. Much more virus were observed in the absence of NLRP3 and caspase-1, but not in the absence of NLRC4. Moreover, a higher lytic effect on viral cells was observed when these cells were cultured with the supernatant recovered from the NLRP3 null and caspase-1 null astrocytes. So thus indicating the role of NLRP3 to control SARS-CoV-2 infection. And as I mentioned before, the activation of inflammasome culminates in a gazermin D-dependent pyroptosis and also cytokine release. So to further understand how NLRP3 controls SARS-CoV-2 infection in astrocytes, we first evaluate the viral load in astrocytes from gazermin D null mice. And we observed a higher viral load in cell lysate and supernatant from these cells, thus suggesting a possible role for pyroptosis in controlling SARS-CoV-2 by astrocytes. However, no significant increase in lactato dehydrogenase levels, that is a signal of lytic cell death, was observed in response to SARS-CoV-2 and neither in the absence or presence of inflammasome components, ruling out the participation of pyroptosis in this process. On the other hand, a significant lower IL-1 beta levels was observed in the absence of gazermin D, in spite of the potential of this astrocyte to achieve caspase-1 activation. So thus suggesting a potential role for IL-1 beta in controlling SARS-CoV-2 infection. In fact, the inhibition of IL-1 receptor results in an increase of viral loads on the cells. But the addition of exogenous recombinant IL-1 beta resulted in a better control, a trend consistent with the IL-1 beta levels observed in those cultures. So higher viral levels, lower IL-1 beta secretion, and in contrast, lower viral loads, higher IL-1 beta secretion. So consistent with this, the addition of exogenous IL-1 beta also reduced the viral load on gazermin D null astrocytes. and assist again in the addition of recombinant IL-1 beta also reduced the levels of virus in the absence of NLRP3 and also caspase-1, again correlated with the levels of cytokine observed in those cultures. So for this first part, the take-home message is that NLRP3 plays a pivotal role in controlling the autonomous ability of astrocytes to control SARS-CoV-2 infections. Our findings demonstrated a classical NLRP3 activation on astrocytes, which culminate in glycerin D-dependent IL-1 beta secretion that seems to act in IL-1 receptor in order to inhibit viral replication. Being astrocytes an important site for viral replication, this early control can prevent the spread of virus to other brain cells, ultimately contributing to the neuroprotection. A potential consequence that needs further investigation through in vivo models that are ongoing in our lab. So let's move to the second part to understand the non-conventional effector mechanisms mediated by inflammasomes to control Zika virus infection. So the question here was, how does caspase-1 control or regulate Zika virus replication by astrocytes? Since we didn't find any classical response mediated by inflammasomes in response to the Zika virus in astrocytes. We didn't observe any IL-1 beta secretion, no LDH release, in contrast to that observed for nigericin. So we looked for some microbicidal molecules, such as nitric oxide, and again, we didn't observe any release of nitric oxide, in contrast to that observed for recombinant interferon gamma interferon. So we moved to investigate the secretion of antiviral cytokines that are important to control, especially Zika virus, and we found that caspase-1 astrocytes secreted even higher levels of beta interferon in comparison to wild-type astrocytes, and similar was observed for TNF-alpha, with no difference for IL-6. Thus, ruling out the participation of this microbicidal molecule and antiviral cytokines to explain the susceptibility found on caspase-1 new astrocytes in response to Zika virus. So, making a very long story short, we turned our attention to the metabolism, since it is well known that Zika virus is able to induce a metabolic reprogramming of host cells in order to uptake nutrients and improve its replication inside host cells. So first, we evaluated the secretion of lactate and glutamate. Lactate is a product of the glycolysis, whereas glutamate is an amino acid produced by glutamine conversion. And we found that Zika virus did not alter the secretion of these products. But interestingly, we found that in the absence of caspase-1, higher lactate secretion was observed in uninfected as well infected astrocytes, thus suggesting a role for caspase-1 in the regulation of glycolytic pathway. In fact, the extracellular acidification rate analysis indicated that in the absence of caspase-1, a higher glycolysis, higher glycolytic reserve, and higher glycolytic capacity was observed in astrocytes, even without infection. And these higher levels were maintained in response to the Zika virus. So to further explore how caspase-1 regulates glycolysis in astrocytes, we explored the expression of genes involved in glycolysis. And we find a higher expression of GLUT1, hexokinase 1, phosphofurantokinase 1, GAPD, lactate to dehydrogenase, higher expressed in astrocytes from caspase-1 null mice in uninfected and infected condition. And among all of these factors, hexokinase 1, that is the first enzyme of glycolysis, was particularly upregulated in the absence of caspase-1. So to confirm the role of glycolysis to Zika virus replication, we inhibited hexokinase 1 and thus the glycolytic flux by the addition of 2GG. And we found a significantly reduction in the Zika virus load in astrocytes from caspase-1 null mice. And this effect mimicked the outcome of wild-type cells, thus indicating the requirement for this glycolytic pathway to Zika virus replication within astrocytes. To further understand the involvement of caspase-1 in the regulation of glycolysis, we performed an interactome analysis between caspase-1 and hexokinase by using this string platform. And we found two completely distinct clusters, and this metabolic sensor emitter emerged as a possible link between these clusters. And in accordance, the inhibition of emitter with the addition of rapamycin again reduced the viral load, Zika viral load on caspase-1, similar as found for 2GG. And we can see here by the observation of intracellular virus, a clear reduction in the addition with the addition of 2GG and also with the addition of rapamycin, thus demonstrating the involvement of emitter and hexokinase 1 and caspase-1 in the regulation of this glycolytic pathway. So the glycolysis result in the pyruvate formation, and this pyruvate can be further metabolized to lactate through the lactate dehydrogenase, or it can enter into mitochondria to participate in the TCA cycle. So in order to further understand how caspase-1 regulated the glycolysis impact Zika virus replication in astrocytes, we inhibit these two pathways. We inhibit the lactate dehydrogenase and the mitochondria carrier. And we find that the inhibition of pyruvate entering in the mitochondria clearly reduced the Zika virus load on astrocytes, in contrast to that observed with the oxamate. So thus indicating the requirement of pyruvate into the mitochondria in order to boost Zika virus replication. And it is in contrast to that described for SARS-CoV-2 infection in human monocytes, in which lactate, but not the transport of pyruvate to the mitochondria was important, thus demonstrating the particular requirement for Zika virus in astrocytes. And to further confirm the role of pyruvate to Zika virus replication, we added exogenous pyruvate to G cells, and we found a higher increase in the virus replication on wild-type cells, but not on caspase-1 null astrocytes that has already this improvement in the glycolytic pathways. So thus confirming the requirement of pyruvate to Zika virus replication on astrocytes. So taking together for this second part, our finds provide compelling evidence of the role of caspase-1 in the regulation of glycolysis through a mTOR exokinase pyruvate axis, although how caspase-1 controls mTOR activation remains to be elucidated. And this metabolic regulation has a beneficial effect, thus preventing the virus replication observed in the absence of caspase-1, in which this pathway is upregulated or dysregulated. So, in conclusion, we can say that inflammasomes boost or drive the autonomous ability of astrocytes to control viral infections through conventional and unconventional effector mechanisms. And this role of caspase-1 in the regulation of metabolism can also have clinical implication, under sterile condition, for example. Given the importance of glycolysis in cognitive function and memory, and such its decline in Alzheimer's patients, and also given the important role, the crucial role of inflammasome in the immune pathology, in the neurological conditions of these patients, caspase-1 can be considered and or can emerge as a possible target, a narrative that I hope to further into that, to delve further in our next encounter. I think it is it. I thank my lab, especially Ingrid Farias, that obtained the majority of data I presented here, all of our collaborations and funding agencies, and please visit us at this website, and let's keep attending this global IMMUNOTALK and following the next global IMMUNOTALKers. So thank you all for the attention, and I'm happy to answer your questions in the Twitter. Thank you all.
Thanks so much, Karina. Thanks so much for this beautiful presentation and this exciting adventure with viruses, astrocytes, inflammasome activation, and metabolism. I think it's a fascinating work. Thanks so much.
Thank you, Gabi.
As usual, this talk will be followed by questions and answers on Twitter, X, and we will now share a slide with instructions. So you need to search for account Global IMMUNOTALKs, then find the tweet that says ask questions for Dr. Karina Bortoluci here, and then reply to that tweet with your question and mention #GlobalImmuno. So thank you very much again, and we look forward to meeting you next week with a new Global IMMUNOTALK speaker, Dr. Daniela Thomen from the Netherlands. So thanks so much, Karina. Thanks so much.
Thank you, Gabi. See you
all
soon.
the audience. See you.
Bye-bye.